Okay, so the first thing that the lab book instructs is to finish filling in that table that we did before when looking at the controls. So I'm going to flip back to that table on page 69 and fill in the last 3 rows. So the silver nitrate still testing for chloride. The initial color is still going to be clear. And the color, it should have come out for you guys, and as we see here in this diagram, so right here, it came out cloudy, meaning that it was positive for salt. The iodine test, which tests for starch, the initial color should be yellow. That's the color of the iodine solution. And the final color, if you look at this, is still yellow. Yeah, it's a little darker than a light yellow, but it's definitely not that black or really, really dark blue purple color. And then finally, the Bendix test is still testing for glucose. The initial color is light blue, but we did see a color change. This isn't as orange as the other one, but it's definitely not blue. So this is saying it's positive for glucose. So, according to our test results, which molecules diffuse out of the bag? Well, looking at the silver nitrate test, salt definitely came out of the bag. The Bendix test indicates that glucose came out of the bag, but the starch test indicates that there was no starch in that water, meaning no starch left the bag. So question 7 asks, if all molecules are expected to move from high to low, why didn't everything move? So starch was a lot higher in that dialysis bag. However, it didn't come out. It didn't go to low concentration. So the reason why is that a dialysis bag is a semi-permeable membrane, and it just wasn't permeable to starch. Starch is a really large molecule. Remember, it's tons and tons and tons of glucose molecules. On the other hand, though, glucose is very small, can move in and out of that dialysis bag. Salt is really small, but starch is really large, so our hypothesis, our guess, is that starch was just too large to move out of the dialysis bag. Question 8 says that the ability to separate molecules is called dialysis, and did we see that happen? And we did. We were able to isolate starch in the bag and get the salt and the glucose outside of the bag. So we were able to separate molecules. Alright, so that finishes the lab activity 4, which was looking at this dialysis tubing. So the last activity, activity 5 on page 72, is still investigating diffusion, but is looking at a different scenario. So in this scenario, you were to make 2 different microscope slides looking at a plant leaf. So when looking at the leaf, we had one that was in culture water, or think of that as just regular old water. And another leaf was in salt water. And so we wanted to see if the cells looked differently between the two. So here are 2 microscope slides of plant cells, and you can clearly see the cell wall in each of these. However, you notice that the inside of the cell looks very different between the two. So here, this image is our plant cells in regular culture water, whereas here are plant cells in the salt. So looking on page 72, you're going to want to draw these 2 cells, have a general idea of what they look like, and then answer the questions on page 73. So questions 6 through 9, as well as the questions on the back. So questions 1 through 4 on page 74. Now if you get lost on answering any of these questions, remember what we're learning about. We're learning about diffusion, and that might help you with some of these problems. So again, using these 2 images, go ahead and complete the rest of this activity. Okay. So first of all, I hope you've already drawn these pictures so that you can go back and study these. It asks you to label your diagrams of the cell wall. So that's this outer portion of each of these cells. The cytoplasm, which is any empty spot within the cell. And then the chloroplast, which in both of these images is the green dots. So in one of the slides, you should be able to see the cell membrane. So in a plant cell, plants have cell walls and cell membranes, and they are right next to each other. Now in the culture water, you really can't distinguish between the two. Here we have the cell wall, and the cell membrane is right up against it. We can't actually see the cell membrane. However, the salt solution, we see this grayish cell wall, but we also see the cell membrane. Remember, the cell membrane is going to hold all of the cell contents within it. So we can kind of see the inside of these plant cells in the salt have kind of scrunched up. And we see this membrane that's holding all of those contents together. So in the in salt picture, you can add a label for cell membrane. So looking at question 8, the control in this experiment are cells in culture water. We want to know the effect of salt water on plant cells. Well, we don't know the effect of salt water if we don't have an image or an idea of what plant cells look like without that salt water. Question 9 asks what's causing this difference between the two cells. So the question is why are these salt cells, why do they look scrunched up like this? Why do they look drained? Well, it's because they are drained. Think back to diffusion. Outside of these cells, so I'm going to draw this out here, we have 20% salt, which means we have 80% water. Now you don't know what's inside these cells. The question doesn't tell you, I didn't tell you, but 20% salt is very salty. You don't lick plants and taste a whole bunch of salt. You probably don't lick plants to begin with, but I'll tell you right now, they're not salty. So it's a really safe assumption to say, okay, there's a lot more salt on the outside of these plants than there is in the inside. Along those same lines, if there's less salt in here, so let's just say there's 1% salt in a cell, that means there's 99% water. So this means the concentration of water is greater inside the cell than outside. So that means the water is going to leave these plant cells, which is causing this shriveled up look because water is leaving those cells, so there's less inside of the cell. This is the same exact thing that happens when you put salt on a slug or a snail. There's a lot of liquid inside of these organisms that leave the snail and leave the slug when there's salt on them. Or if you've ever been to the beach and your lips get very salty, it's the same thing. There's a lot of salt in the air, so the water in your lips is going to leave in order to make an equal concentration of water on the inside versus the outside. Alright, so flipping to the back on the comprehension check on page 74, this is saying that let's estimate that the inside of an Elodia cell is 98% water. So the salt solution you apply to the slide is 80% water and 20% salt, because remember this is a 20% saline solution. Using the information in question 1, add an arrow to the picture you drew of the Elodia cells in saline solution showing the direction of water movement. So water was inside the cell and it's moving outside the cell. The process that occurred in the Elodia leaves is an example of osmosis. Now recall, both of these are passive transport. However, osmosis solely deals with water. Both osmosis and diffusion are moving from a high concentration to a low concentration, but anytime we're talking about the movement of water, that solely means it's osmosis. If water is moving, it's called osmosis. So looking at question 4, it says a student performed the same experiment on human blood cells. They put it in salt water, they shriveled up and died. When they put it in distilled water, they filled up and exploded. So what organelle that's present in leaves leaf cells, but not blood cells, prevented the elodea cells from shriveling or exploding during your experiment? Well, one of them is the cell wall. The cell wall is a very rigid structure. As you can see here in the shriveled up plant cells, the cell wall still exists. It can refill back up. Now, another organelle that you may have come up with is the vacuole, the central vacuole. So you can recall this from the activity you did in lab, actually in person on Tuesday. But also, if you flip to page 73, look at that hint that's at the bottom of the page. It says that there's a large central vacuole that functions in water storage. So meaning, when these cells that are in salt water, when they're losing water, they still have this central vacuole that has a water store. Or if you have plant cells that have tons and tons of water, instead of it kind of bursting out the cell membrane, it gets stored in that central vacuole. So the central vacuole is just acting as another way to balance the water. So both the central vacuole and the cell wall are both correct answers to question 4. So that's the end of the lab activities. Remember that you are responsible for all of these activities. If you have any questions at any point, please just send me an email and I'll help you out as best as I can.